We welcome you to the IMAG MSM working group weekly meeting on multi-scale modeling of viral pandemics. And I need to remind you that this meeting is live streamed on YouTube, recorded and made available publicly. So please be aware of that. We have a single talk today by Nancy Baker, Center for Computational Toxicology of the EPA. As always, you have myself, Tom Helikar, who are here to take your suggestions, comments, and complaints. Jim Sluka, if you have anything you want done to the website, you have great things to post there, please contact him. Lorenzo, who can't make it today, and Joel have been doing an incredible job handling the activities. And we thank them for that. So these are the emails that people need if you want to contact us. We have our various communication channels. Please do help us use them. We have the Twitter hashtag MSMViral. If you tag things with it, that would be great. If you want to post to any of these feeds, let us know. Please reference our great archive of talks on YouTube, and we would be grateful for your help on that. Again, if you can follow us on Twitter with that MSMViral hashtag. Are there any announcements before we get to our talk? We do not have a digital twin subgroup meeting tomorrow because Reinhard is on holiday. We will not have a working group seminar next week. As usual, I do ask you to help us suggest speakers and fill up the schedule. John, go ahead.
Just a quick question. The Slack account that we have, is that a paid account? Because there's a notice out there changing their policy on how much stuff they'll let you keep on there before they start deleting.
No, we are not a paid account. We are on the free account. And as expected,
Okay, yeah.
the social media starts off free.
Yeah, it was some fairly large number of bits, but I think they're going to a time thing. Anything over some age, they're going to drop out. So might need to check that. That's all.
So we probably need to archive that so we have copies. Thank you very much. Any other announcements before we move on to our talk? So schedule at the moment, we're going to have our summer break next week. We have Carolyn Cho from Merck on the 25th. We have a number of speakers lined up in the fall, but there are also plenty of open slots. As always, you've heard me say it many times, suggest yourself, suggest your students, suggest your postdocs, suggest the people whose papers you like. Think widely. It could be people doing methodologies as we're having today. Could people be doing experiments, clinical drug development, anything that you think would be interesting to hear and that you think this community should have access to. And I'm now going to come to our talk and turn over the introduction to Nancy, to Tom, and then we will have our seminar.
Great. So, pleased to have Nancy Baker speak to the group. Actually, after Caitlin and I spoke last month and talked about some of the work we got out of her tools, this group asked to hear from her, so she's kindly agreed to speak to us. Nancy is a contractor with Leidos. As you might know, Leidos is the contractor that maintains PubMed. But she's been working at the EPA in North Carolina for at least, I think, 10 or 11 years, where she's been working with my team on the virtual tissues. Nancy got her degree from University of North Carolina in information science, and she's had experience at working at local drug companies, and we were able to bring her into the EPA for her expertise in information retrieval. And she's developed a tool called Abstract Sifter, which is in version 7. She gives lots of tutorials, which you'll give to the group. And I think you'll see that the functionality really extends what you can do with PubMed. And she's always happy to provide video tutorials that she's prepared that are accessible. And there are versions of this that are on our EPA CompTox dashboard. So most of the work, or most of the tool is publicly available, but she's got new and exciting things that she's got that are not yet publicly available. And she'd certainly appreciate feedback from the team in terms of ways that the functionality can be expanded and tailored to be more useful to the needs of this group. So Nancy?
Thank you, Tom, for that lovely introduction. And I will try to share my screen here. Let me know when you can.
Go ahead, you have host privileges.
Okay, so you can see my screen now.
Go. Yep.
Okay, excellent. Again, thank you for inviting me, and thank you, Tom, for that introduction. And yeah, it's going on 11 years that I've been working with Tom now. And the kind of work that Tom and his team do is the kind of work that really puts a stress on getting information out of PubMed. So that's why I developed this tool, and so it was designed specifically for these very headache producing complex biological searches. And Tom has assured me that you all are very interactive and will jump in and interrupt me at any point, which is what I really like, because if you don't interrupt me, I feel like I'm talking to myself, especially on Zoom. And I'd like to acknowledge Tom and the whole virtual tissues modeling project as it's evolved over the years, and then the support of the CCTE management and the scientists. And of course, as Tom mentioned, the Abstract Sifter and users have been giving me feedback over the years, and that's been really helpful. And of course, the problem, of course, this challenge with any sort of big, can you all see this thing? Do I need to get that out of the way? Okay. The big challenge with working with complex biology and big biological areas is there's just so much out there. And I just did a couple of queries on different topics that seem maybe you guys were interested in. And it's just, you can't get anything that's under a couple thousand articles. And then, of course, you get a couple thousand articles and you have a couple thousand articles. And then, like, what do you do then? So there's tons of literature out there, and there's so many subtasks once you have identified the corpus you want and getting whatever it is, the information you want out of it. Yes, okay. So this is an example of a project we worked on the last couple of years with the RIVM group. And this is, I'm sure, very familiar to many of you, where you take an area of biology and try to figure out all the major players in that biological realm and how they interact with each other, and then you have to try to make sense of it. And there's a lot of work involved here, and this is an area that the Abstract Sifter can really help you with. So today I'm going to go through just like 4 major steps that seem to be important to all of the kind of projects that Tom works on, and probably you will find very useful as well. So the first one is how you can use the Abstract Sifter to just take some biological corpus, like you do a query on some biological area and you get a couple thousand articles, and then you want to know what are the players, who's involved in this biological area, what genes and proteins, in other words, are involved in this area. And I say it to do this in an unbiased way because I'm going to show you how extracting the mesh terms from those articles is a way for you not to like come to the problem with your own list of genes in the back of your head, but to look and see what what curators and annotators have discovered in this corpus over the years that corpus has been building up. And then you can go and examine that. That's what I'm defining as unbiased. And then I'm going to show you how you can take this complex list of players, genes, proteins, and find out how they're related to each other. Do they co-occur in articles, which is generally speaking, the way you identify a relationship between two genes is if they co-occur. This is sort of the text mining paradigm, that things are related if they co-occur. There's more likely if they're related if they co-occur in lots of articles than if they don't co-occur in lots of articles. And then you can look at that in your biological context, but you can also look at it in some other biological contexts. For instance, you may be looking at embryonic development, but if two genes co-occur outside embryonic development, maybe that's significant as well. And then number 3 is take that your set of players, genes, proteins, and then measure their co-occurrence in some biological corpus, maybe the one you're interested in, as I said before, maybe some other one. And then, and this is usually the end point of what we do, is you want to know, you built your pathways and you now want to perturb your pathways with some environmental chemicals or drugs. And so you need to develop a list of chemicals that you know are likely to perturb some target in your pathway. How can the abstract sifter help you with that? So I will show you very quickly how to do each of these 4 major steps. And as Tom said, the abstract sifter is a free tool. Right now, this is the download link, but you have been emailed, I hope, the newest version, which is version 7 with the version 7 user guide. There are new versions coming out, but we're putting some processes in place that have tended to slow that down. So the version on the dashboard is not version 7 yet. So you have that, though, in an email. And I hope, unlike Slack, you will never have to pay for the abstract sifter. So let's hope. Okay, so I'm going to get out of here and go into the abstract sifter. The abstract sifter is an Excel macro-enabled spreadsheet. And I have opened it up here, or had it open. I don't know how to move this thing here. Down here, maybe. Okay, I have opened it up to the main sheet. There are many sheets in the abstract sifter. Main sheet is the one that is most reproduces what happens in PubMed. You send a query, you get the results back on the main sheet. There are a number of other sheets that we'll go through today. The user guide that you should also have in your email will walk through each sheet and what the specific functionality of each sheet is. But so let's start with the main sheet. And there's this one button here on the top, says Query PubMed, and here is where you put in a query. And I've already run this query, so I'm not going to run it now. But what happens here is you put a query in and you click on Submit. And then this query through an API call is sent to PubMed. It's resolved, and the records are sent back to Excel, and the abstract sifter code behind the scenes takes each record and writes it to one row on the main sheet. And I think this is like 1,600, 1,800, yeah, 1,600 citations satisfied this query. So they're all right here, which in itself is nice because you can, this is hyperlinked PubMed ID, you can read the title, you can look for authors, you can use the built-in Excel features to search on certain things. And that's nice. You have all of, okay, how do I get rid of this thing? Put this up here now. You have the usual links for DOI. You've got the journal name, volume, issue, etc. And you can sort it any way you want to. This particular corpus, the earliest articles, 1952, and of course, there's tons of articles in 2022 that fall into this corpus. So there are lots.
Excuse me, Nancy.
Yeah.
Can I, since you're expecting us to jump in?
Jump in.
So, but you broke blood and brain into 2 queries based on what was input?
Okay, so my query was blood-brain barrier. Okay, are you talking about these cells here?
Yes, and
Okay.
do you need to quote phrases in
No.
your query?
Okay. I thank you for setting that up. Okay. So these three cells right here are what are called the sifter cells. And you can put, they're not, I'm not going to call them search terms. I'm going to call them sifting terms, because what you put in here is, I'm going to put placenta, or the part of the word placenta. So what happens when you put these characters in here is that the abstract sifter takes that string of characters and counts the occurrences of that string of characters in your titles, abstracts, and keywords. So this particular article right here, or citation, it's not the whole article, obviously, it's the PubMed record, has 14 occurrences of placenta. I could put
Okay,
egg.
so those are terms from your corpus that you returned. But my question is in your query, how do you do a noun phrase? If I do estrogen receptor, I do not want all the abstracts with estrogen and all the abstracts with receptor.
Well,
I want the abstracts with estrogen receptor.
okay, so that's a PubMed question. You would probably just put quotation marks around it, or you would
Right.
find the mesh term for estrogen receptor and just put that in, and then it won't break it apart like that.
Okay,
Yeah. So, and oftentimes when I have questions like that, I will just go to PubMed and type it in real fast and see what happens, because then they'll tell you if something's wrong, or they give you more feedback than the abstract sifter can give you. But definitely learning how to do good queries or different queries, specific queries in PubMed is something that there's a user guide for in PubMed, and I spend time on there myself. I'm not an expert, but writing the correct query is important, because sometimes you get way too many articles and you could easily just change your query and get a better number.
Yeah, Jim, she can write very complex queries and dozens of them to populate these sheets. So that's part of what she relies on in working with domain experts, is to get the right strategy for your queries to capture as broad a corpus as you want.
sure. I mean, it gets worse. I mean, estrogen receptor, if you want to talk about, there's 2 genes in humans, and each one has about 5 different names. And so how do you generate? Well, it's estrogen receptor alpha or estrogen receptor 1, or ESR1 or ESRA or ERA or ER1. So it can be a tremendous challenge.
And then you probably end up with so many articles that I could try at the end. Let's play with that for a little bit if we have time, because that's an interesting
Okay.
question.
Okay.
So my example here is I've run a really simple query to PubMed, but it's very broad, it brings back just the right number of citations for a presentation. Normally, when I work with Tom, we might have 5,000 or 6,000. This is only 1,600, which isn't bad. But these sifter cells will help you find from the 1,600 or 5,000, however many you have, what you're looking for, what is relevant to you. This is an information term, a science term. What is relevant to you at that minute? So if I'm thinking about embryos and so on, I can sift on these specific terms like this and look at overlap, and then maybe I'm going to look at egg. I know eggs in there. And search and sort by sift and sort by any number of terms, and I can go on like this all day. And let's say this is maybe glial. Maybe I'm interested in glial. Now, when you find an article that looks like it might be interesting by knowing which sifter terms it has, you can then double click on. Let's do one more. Let's do blood. You can double click anywhere in the row, and it takes you to another sheet called the abstract sheet. And that's where you can actually see the abstract. And the terms that you were sifting on the main sheet are colored in the same color they are in the main sheet. So here I've got glia in blue, blood in, see that, blood in green, and embryo in red. So my eyes can quickly pick out those strings of characters, those terms that I was interested in, and I sifted on in the main sheet. And then I can, just keep on going. I want to look for macrophage. Let's do it. Keep, sift and sort as many times as I want to. People always ask me though, what if I have 5 terms I want to look at the occurrence of, or 10 terms, or what if I have a list of 15 genes that I'm interested in? How can I look for all those at once? And that's what I'm going to show you in a little bit. So keep that in mind. So that's the basic functionality of the main sheet, is to do queries, get your corpus, and then using these sifter terms to find things that are of interest to you. I wasn't going to show this today, but real quick, if you do find articles that are interesting, you can say, let's say this is good. These are all interesting. I want to go get the PDFs for these. You can take group notes and put some sort of tag in there, some sort of, I'm not going to write anything because that's would take too long, and then say, okay. And what that does is that it writes these records to the note sheet. That's these. I'm going to clear off what I had there before. And now when I say I could save this abstract sifter and I come back tomorrow or next week, I have taken notes on these. I don't need to look at them again. Maybe I've already gone and got the PDFs or whatever. And these are ones I want to dive into. Okay, so the first thing on my list here today to show you was, okay, you've got this big sort of fuzzy biological area, and do you want, in a non-biased way, that is some expert telling you what are the players in this corpus without you having any opinion about it beforehand. Maybe you have a student, you say, okay, go get to know this area of biology. So what I'm going to show you next is this facility, this capability where you can extract the mesh substances. And I'm going to go ahead and get all of them, because right now I'm interested in the genes and the proteins, and I want when I run this little bit of code here, what's happening is that for each one of these citations, the mesh terms, the mesh substance annotations, these are what the National Library of Medicine attaches to articles, are all extracted and organized on this sheet. And so I have 1,600 articles, and I have over 2,000 substance terms. So I've been brought to a sheet called Mesh Mine, and here are the mesh terms that were extracted from that 1,600 articles. And I can sort by the name. I can sort by the article count. Maybe I want to find what occurs in the most articles. I can also sort by whether the substance is described as being toxic. So this is be interesting for environmental chemicals or drugs that have side effects. I can also find drugs through whether or not they're associated with the therapeutic use, and whether they're family terms. Family terms is something I usually clean off first, because I want specific entities, specific genes and proteins. And then over here are the different categories of substances. And in this case, I am looking for gene and protein players. So I'm going to turn this flag on by just sorting by it. So I'm only looking now at genes and proteins and amino acids. And then over here are my genes and proteins sorted by the number of articles that they're in on the main sheet, and the 1,600 articles on the main sheet. So this is where you can start to get a feel for what players are in your particular corpus. And you can see that some I have a really high count, like this ATP binding consent transporter is got 42 different articles, but there are lots of things that only appear in several articles. And we get lots of different, very specific genes here, even down to what genes in specific species. So you get this list, and then you can start exploring it. Well, the first thing you can do to explore it is these are all belong to the mesh vocabulary, which has its own dictionary. So if you don't know what something is, you could say, click on it, and that will bring you over to the mesh library browser and tell you a little bit about it. And so you can do that. And then also, if you want to see the articles, you can just double click over here. So let's look for Claudin 5. what all that does is writes Claudin 5 on the sifter cell and sorts. So this particular article here has 7 occurrences. So these are all these are all the articles that have Claudin 5 in them. And then this particular one has Claudin 5 7 times in the title and abstract. So it's the richest in it's the richest in the term Claudin 5, and you can see it has a lot of Claudin 5 in it. So
another interruption and quick
Yeah,
question. So
yeah.
so will it capture the ones that have Claudin 5 without the dash?
That's a really good question. Okay, so these are mesh terms, and let's see here. Let's look at this one. So you can see that the mesh term here has the dash in it. So if you have that question, and that's a really good question, then you could just write here, Claudin 5, like that. And you're right. So here's one that has it probably as the mesh term with the dash. Yeah, the mesh term has the dash, and then whoever wrote this article did it their own way without.
So there
the
isn't
dash.
actually any way to get Claudin space 5 in Sifter.
Okay, say that again.
It's Claudin space 5.
Oh, space 5. Yeah, okay, sorry. Yeah, I can do that. Claudin space 5.
Yeah.
See how many we have here. Yes, here's Claudin space
Yeah,
5 right
okay.
here. Yeah. And that's a really good point, because as you know, I'm sure every gene has so many different spellings. Every
Yep.
chemical has so many different spellings, and then people will abbreviate it in their own special way. And you don't always know what it is. So yeah, so we have a nice assortment here of different ways of going after Claudin 5. So keep that in mind for the next thing I show you. Okay, so oops. So let's say whoever it is, your student has identified some interesting players here they want to do more research on. Let's take these 3. So and that one, and here, let's see here, maybe these 2. I'm going to copy these and go over to the landscape sheet, which is another sheet that lets me get an overview. I'm going to paste them here. And I already had something out here from playing around earlier. The landscape sheet is a very simple grid. So column C is where you put some terms that you want to query. And over here along row 3 is where you put other query language. And I did enter a query earlier, embryonic development, and I ran it against these genes. But what you can do with this sheet is you can select cells. Well, let me do it like this. You can select a cell, double click on it. What happens here is that Desmond and embryonic development query string gets written based on whatever is in this row. This is the cell we clicked on, and then whatever is in row 3 on this in the same column. So if I just select this cell and say update article counts, that query that we were just looking at gets run and sent to PubMed, but I only get the number of articles back. So now what I'm getting, the information I'm getting here is that I've identified these proteins as being interesting to my project. And now I'm going to go out and find how many articles in which embryonic development and my genes or proteins co-occur. And I can then visualize it. And then I can modify this any way I want to. Let's say this is too broad, this is too narrow. I want to broaden it out to any species. I can modify it. I could put the long name here of the protein next to it. I can't remember what this protein is. So I can start to get an idea for each of these entities, what their occurrence is in this embryonic development, along with embryonic development. I can also take some of these proteins, copy them and put them here. And I'm going to format that nicely. Okay. And so now I can get the co-occurrence of these proteins. Well, here, I'll just give you an example. OCLN and matrix metalloproteinase 2. I can get the occurrence of that. I'm not going to run it, but just by selecting the intersecting cell and then clicking on update article counts, the queries will be constructed iteratively and then sent to PubMed, and the number of articles, well, that's the same as that. It's number of articles sent back. So you can see that I can take this sheet, and sometimes people create really large sheets, and I can look at the co-occurrence of my players with any other terms that I can look up in PubMed. I can also, I have some sample queries here. I can look up, say, heart development, heart cells. These are just queries that I have written. I don't know why that. And that's a very complex query about heart cells. And now I can look at see how this is a much more complex query. So now I'm looking at interleukin-2 and cardiac heart progenitor, so on.
So that would be great if I could maybe we could have, I got distracted because I was trying to run it and my computer was saying macros are not enabled and so on. So I got distracted, but this is fascinating. Maybe one or two people could suggest a query you could run. I mean, you've got interleukin there.
Sure.
Could you try interleukin, interferon, and steroid?
Interleukin 6, or
Do you have to give a number, or could we just do
No.
IL or interleukin?
it might be a lot. Okay, and what do you want to know about interleukin or interferon?
Well, I was going to say, I was thinking interleukin, interferon, and steroid, because I'm interested in mechanisms of how steroids suppress the early interferon response.
Okay, so this would be the nope, here it is. This is the intersecting cell with that and that. So that's the query. And if we just click on it and go get the article counts. So 22,000.
Yikes. Okay, so I have to be more specific.
Well, yeah, so there is a limit to how many you can bring back on the main sheet. So that's a bit many. You could, for instance, like interleukin 6, you could go, and then interleukin, what is there, an 8?
Well,
and then you could give
try
me a little-
IL-1 alpha and IL-1 beta.
Come on. Okay, we're getting down there, or IL-6. IL-6.
And with the interferon, we could say type 1 interferon, although I'm not sure what the correct search term would be.
You're going to spend a lot of time getting interferon to query correctly. I would change steroid to say glucocorticoid.
That would be better. But we could say fluticasone specifically.
They cut it in half.
Fluticasone, that's what I use when I have allergies.
Don't.
Don't? I used to work for Glaxo when we were making the stuff.
I stopped taking it after I read the last couple of articles.
So this is how you can get visualize the overview, and then to actually read the articles, you just double click and then say submit, and it will run the query and put your articles on the main sheet. But it also deletes. I can append it to the main sheet, but in this case, I don't want to get rid of what's on the main sheet because I might still need it. In fact,
Does anybody else on their call have a query they'd like to see that might be relevant to something they're doing? So this was helpful to me just to see this little demo.
Well, the other thing, will make your secondary column drug. So just do like IL-6 as your row and drug as the column. that basically you're trying to find, are there drugs that interfere with IL-6 or upregulate or downregulate? You really don't care, but it's, yeah.
Yeah, I think maybe something,
Well, interferon, interleukin, all by itself probably returns 1 million articles. There's like 35 million articles in PubMed. So, I mean, that's a grossly, so Claudin 5, but those Claudin 5s, okay, there's two of those numbers the same. So somebody's searching dash the same as a space, presumably PubMed.
yes, they may be. Yeah, that's
But that would be a
possible.
start to find it. If you found Claudin 5 in something and you want to know what can I throw in my assay to do something, that's a start of how you'd go about doing that.
Well, good. So you guys, I think you're getting it. So
Yeah.
this is, yeah,
Yeah.
you put what you and you futz with it until you get it to be what you want it to be. Just remember that it's column C and row 3, and this intersection area will build the query with what's in those two regions. And then with the sample queries, you can pick from for quick, more complex things. And if your group has queries, you can share them and save them on the sample queries. Just add them to the bottom of sample queries and share them with your colleagues. Okay, now I'm going to go back to the main sheet. And so we talked about well, let me just recap. With MeshMind, we looked at what the annotators at PubMed have collected for this 1,600 articles over the last, I don't know, what was it, 60 years or whatever. Now we're going to look at, say, you have a set of genes that you maybe test for, and actually, maybe I'll show this first. Maybe you have a set of genes that you know how to test for, or you're interested in, or whatever it is. So we're talking a little more biased. This is something you want to see. That's where another sheet called the term mapping comes in. And the term mapping means that you specify a list of things you want to sift on, basically. So maybe you're looking for rodent studies. Let's use that as a quick example. And when you're on the main sheet, you might put mice here, and then you'd sort on mice, and then you might sort on let's go rats, and then you might sift on that, and then you would go, okay, what about what is it, F fish or whatever it is? I can't remember what it is right now, or rabbit, or whatever. You would go back and forth, and then you'd look for MIGs because you're looking for dose studies. So there might be a whole bunch of things you're going to look for, and you only have 3 sifter terms. The term mapping lets you have lots of sifter terms. And so you could, if you're looking for dose studies, you could put dose, you could put MG. I don't think that's in there now. And then studies. So the basic layout here is that you put what you want the sifter to look for and sift on, kind of, and then you map it to something else. So when I put these here and I click on the map, what the sifter is going to do is going to look for this string of characters, and sometimes they have spaces in front or in back, and then count the articles, the rows on the main sheet, the citations on the main sheet that have that. So for instance, there's 106 articles that have Sprague dash Dolly, and 3 that have just plain Sprague Dolly, and we have Long Evans, and so on. And to see it, just you can double click here. It puts it in the sifter column here. It sorts. Or if you're interested, if you really want an article that's about rodent studies, if you had 1 of these terms, it's probably about rodent studies. If it has 2 of these terms, it's more likely to be about rodent studies. If it has 5 or 6 of these terms, that's an article about rodent studies. So if you want to find the articles that have the most of these terms, you double click here, and this first one has 11 of the terms there, which is quite a few. Let's see what it says. So we have guinea pigs, rabbit. Okay, I did put MG and MB instead of MG. Guinea pigs, mice, mouse, MIGs. And why is MB there? Guinea pigs. So this is definitely a article rich in rodent stuff. So I'm gonna get MB, I'm gonna get rid of that. That's not what I wanted to do. I wanted to do MG. map that again. So you can imagine, I hope you're all thinking, oh, I have a list of genes from a pathway, and I could put them there and then see how many of my genes are in my corpus. And that's what we're going to do right now. We're going to look at this totally made up, 2 different pathways, totally made up, make no sense at all. I'm just going to use this to show you the mechanics of this. So I'm pretending that I have, why did it do that? I'm pretending that I have a pathway I've defined so far as having these genes, and I put a couple variations in their names because I don't want to miss any. And then I've got path 2 that has some other genes in it. And I want to see how many of my genes, the ones I can test for, the ones I'm interested in, are in my corpus on the main sheet. So I put them here and click on map. And I can get the individual counts here. So there's our Claudin 5. Has lots of Claudin 5 in it. And I can also click here. Okay, come on. And I can find the articles that have the most genes in path 1. And that's this article here that has FGF2, glial, this thing, TJP1, blah, blah, has, and you can look down here at the bottom. This tells you how I actually do this particular functionality, is that I add this new section on that has the lower level terms and then attached to this higher level term, in this case, the path. So I have TJP1 in path 1, FGF2 in path 1, fibrous growth factor path 1, and I also have from path 2, I also have glial fibrillary acidic protein, and also from path 2, zona occludens. And if I put TX path 2 here, I can see how many genes from path 1 versus path 2. Yes, is there a question?
So I was going to ask a question. So let me try to understand how I'd use this. So I have some commercial assay that does cytokines. And now I run my experiment and I notice that there's significant differential expression in the following 8 cytokines. I would now put in the list of my cytokines and some correlated outcome that I'm interested in, and that's how I would use it.
Yeah. So you could put anything here, XXX, and then it's what, cytokine 1. I'm just making this up. Cytokine.
It might be a sort of IL-1 beta is differentially expressed. IL-6 is differentially expressed. VEGF-A is differentially expressed, and so on. And so I want to know, I'm trying to figure out how those things are connected functionally. And so I'm trying to look at the literature from what my experiment has shown me to try to see if anybody's understood why they're related.
Yeah, so in this case, if I put IL-1, I could also, you can't put an or here, so I would put interleukin 1 here, dash on whatever, and then put any other, I'm sorry about the colorization, I'm going too fast here. Okay, let's see what happens here. Yeah, so you build up your collection of genes, and then if you want to see which article has the most of your genes, you double click there, and this should have, here it says IL-6, IL-1 beta, and somewhere interleukin 1. Oh yeah, right there. Okay, there, got it.
It's the IL-1 beta, so it's pulling up from that. Okay, because I mean, as an example, the co-occurrence of VEGF and IL-1 wasn't obvious to me. Retrospectively, I can understand, well, it's a vascular permeabilization during infection, but it's not immediately obvious that would be the case. And so finding literature how that connection would be interesting after seeing it in the experiment. So I think this helps. Yeah, this helps a lot in terms of.
Yeah, I don't know if we got any VEGF there. Whoops.
What can you do with the corpus of abstracts?
That third line sounds promising. Pro-inflammatory cytokine chemokine release, mask of my macro. and then vasculature. So that would be great. So,
Are there
Go ahead.
anything that you can you it so you've now got a corpus of abstracts, however many abstracts this particular query returned. You'd like to be able to do things like, I mean, word frequencies are interesting, cloud count is interesting. The problem is nothing in biology uses one word. There's literally nothing in biology that's a single word. Everything's a phrase. Estrogen receptor, inflammation, drug-induced inflammation, et cetera, et cetera, et cetera. They're all phrases. So are there any tools to help analyze the corpus for common phrases, or even just doing the word count in the corpus?
That's her
I
next
think that's
point.
what your keys are doing.
Yeah,
your
that's her next point.
Okay.
Well, okay, let me show you another example here that might be useful. So we have a lot of people that study environmental fate of chemicals, and they have a lot of phrases too. And so they'll often, when they do their queries, they'll often put these long, 3 or 4 word phrases in to pick up certain articles. And my point to them is go get if you're studying a chemical, just go get everything about the chemical and then map parts of the phrase to your corpus. So get a big corpus, and you can build the corpus very large because you can append things to it, and then so you can run some query and then append results to the other query and keep appending till you get pretty large, and then you can also delete any duplicates. So you could have 10,000 articles here, and then if you do have phrases and variations on phrases, you can this isn't the greatest example, but like fate and transport, you could put them here, and then as you go along, you're going to be discovering new ways people express things.
Nancy, I think what Jim was asking is, once you have all of this, how do you dig deeper? So going into the notes, for example.
Okay. Yeah, so we talked about notes earlier. Let me take some notes here on this, which was the one that Jim was interested in. I'm going to take group notes on that one. So if you think you're interested in something, you take it and you make a note on it, and you've got it on the note sheet, and be sure to save. And the notes sheet, also double clickable like everything in the Abstract Sifter. And when you open up this is what I call the curation form, so you can curate your notes, and you can also read the abstract here too. And Tom uses this a lot, because oftentimes what we're doing is we're reading many, many, many abstracts, taking some piece of information off of the abstract, and then maybe weaving that piece of information into a review paper or the review that goes with our paper or whatever. And so if there is something you're interested in, like here's a list of genes, blah, 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 you can select a piece of text and just drag it over like that. And then you can also attach a local PDF. You could open up the PDF and copy text from the PDF and paste it here. And there's also this is an area I'm building on. There's a data curation capability. So if you want to go to the PDF and copy tables, and actually there's going to be an automated way to do that, or copy, cut and paste figures from the PDF, this sheet is specifically for one article. So you can start to accumulate information here for that one article.
Can you go back a tab to the one you're on before?
Mm-hmm.
That one. It would be really handy to have a 3-slot box there that's subject, predicate, object, so that you can make an XML or a ontology language statement saying that estrogen receptor or estradiol binds to estrogen receptor, and it's an RDF triple that you can reason over.
Okay, so I don't have that yet, but until I do, you could do like this, binds and then whatever.
Yeah.
And what
estrogen receptor.
Estrogen.
I mean, that's a trivial one, but it could be, drug X is used to treat disease Y. And now you're actually building up a knowledge structure that you can use reasoners over.
Absolutely. That is a great idea. And I don't have it built in, but you can build it in yourself and then just use this column here as data.
Yeah. Well, you'd like some help because the challenge is, for instance, you'd like the predicate to come from a finite list of predicates, defined by the relations ontology or something like that. So there's, part of, involved in, leads to, binds to, da 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 da. Just to keep things consistent. But that would be really handy. Well, yes, but I'm still interested in what you can do. You have a corpus now of, well, however many, 20,000 abstracts for this query. Obviously, it looks like you could export the titles and abstracts for all the documents. So if you had some external text mining tool, I assume that, you could save a text file and ship it off to wherever you want it.
Yes, and there are a couple things coming. One is that I think it's 7.3. You can export the main sheet, everything on the main sheet, to RIS file, which is what is a very common, it's like what EndNote uses. Right now, you can, any of your notes, you can export to RIS format. So that, and that's just the citation information. You can also export to a text format. Let me show you what that looks like.
Actually, you might not. Yeah.
So one of the things that Tom does is that he'll, take all these notes and then go into his manuscript and do that copy that I just did and then paste. What? Okay. Paste. So if he takes big, and I did not take very good notes here, but he takes really voluminous notes where he actually is writing, and then he can delete all this stuff, and then he's got exactly what he needs to put into EndNote, and then his other stuff, and then he weaves that into his manuscript. So that's one way to do that.
So I'm sorry, so I don't actually, there's no way to get your corpus exported, but can you export the PubMed query that regenerates the corpus?
Oh, sure. Oh, yeah.
So you actually, because then you can use PubMed to give it, all your abstracts in a TXT file.
Yes, definitely. And the log sheet is another sheet that's really important because that keeps track of every query you've run. So if you want to just copy this query and any of your queries and go to PubMed and run it there, you can do that. I mean, you could do that.
Okay, so,
you don't need me to do
this
that.
This is important. You need to be able to export the corpus of titles and abstracts.
Okay,
one way, by hook or by crook. It's a capability that's needed.
Right. So now with this version, you do that from the note sheet. But if you want to do your entire corpus on the main sheet, you can either just send this to PubMed and do it there. In 7.3, I have a way to write to RIS from the main sheet because you're not the only one who's actually also import from RIS too. I had somebody ask me that too. Can I import? I have, like a couple thousand things at EndNote, and I want to be able to sift on them. Can I import them from EndNote? And that's capability too. So to and from RIS. But of course, the other thing too, I mean, it's obvious here is that this is just Excel. So there's the PubMed ID, there's the title, and the abstract is just a hidden column here. Right there.
Okay, so you could just copy and paste and put it in Word and put in the line breaks where you need them. Okay, that would be good.
Yeah, it's all here.
Yeah, but I mean, there's a ton of very sophisticated text mining tools, and when you're starting to talk about corpuses of tens of thousands of abstracts, it'd be nice to be able to get the data into those other analytical tools. The things that I would like to see, I want to see a phrase analysis. I want to know what phrases are in this document corpus. I want to do a word cloud just because everybody loves word cloud. It doesn't actually tell you anything, but everybody loves their word clouds. Right? You could probably put a tab on here that does a word cloud.
I'm sure Excel can do word clouds. Yes.
Yeah, I'm sure there is another one there somewhere. I would like to do real proximity, sentence level proximity. Because right now your proximity is the first sentence has the phrase and then the last sentence has the second phrase, and they're so uncoupled from each other. The relationship is questionable. So I'd like to be able to say I only want proximities in the same sentence. I want to be able to do things like say, given this corpus, find all sentences with the word pathway in it, and then extract the name of the pathway from those sentences. So that's why I had asked about putting path in it. If you just highlighted path in all your abstracts, you could at least just start flipping through your abstracts and it would highlight the word path and you look around it and say, what
so you're
pathway
talking
is-
about like a regular expression that says, okay, look for pathway and
Yep.
find anything like right
Yep.
before pathway.
Right, whatever, up to the beginning of the sentence with, some kind of way of delimiting it, so that it's, so I can find those a little bit more. So extracting knowledge out of the text itself. It's also possible, of course, that that's in the mesh terms.
Well, they do a lot of that mapping, but you really just need to read and look and see. Yeah.
Okay. Well, see, the reason I'm bringing this up is I had a company for about 10 years, and this is exactly what we did, except we didn't do it in Excel. But it was based on the idea of an omics experiment generates a list of 300 genes. What do you do with a list of 300 genes? And you said at the very beginning, you search one gene and you get anywhere between 50 and 50,000 articles. So what do you do with 300? It's even worse. So the idea that just like you're doing here, so what we want is all the documents that have at least 2 of my queries in them. And then refine it a little bit farther and say, well, the queries have to be in the same sentence. But then you get a breakdown because the query is estrogen receptor 1, but the abstract says estrogen receptor 1 parentheses ESR1, which breaks all your proximities in the rest of the document because you got a local acronym. So you have to deal with the local acronyms and pseudonyms.
Mm-hmm.
Given this corpus, find all the phrases that look like they're the name of pathways. Given the corpus, let's find all the noun phrases that look like noun phrases. So you find a term like estrogen receptor, which is a phrase and not a word. But this looks very good and it's still supported, which is a big advantage.
So when you talk about pathways, is there a ontology of pathway names?
There is, but nobody uses it. Just like there's an ontology of gene names and nobody uses them, right? There's an ontology of tissue names, nobody uses. There's an ontology of cell names, nobody uses them. So that would be another, tool to consider here is when somebody enters in a query interleukin 1, to have it be able to do a lookup and say, well, according to gene cards, it's called interleukin 1 alpha and interleukin 1A and with and without dashes and, et cetera, and just pull all the pseudonyms out of gene cards.
Okay, so mesh does some of that. So let's look at interleukin. Did you say 1?
Yeah. Well, actually, I shouldn't return anything because there's no gene called interleukin 1, but I'm sure there's 5,000 articles in Medline that say interleukin 1, even though.
Well, okay, so here there is an interleukin -1, and it's got its entry terms. That means things that mesh or that the programs will look for to map to interleukin 1 are these here. So IL-1, interleukin.
Right, and it's wrong, right? Because there is no such gene as IL-1. IL-1 is a family, not a gene. So, I mean, you always have this problem, and it's a common problem. But we'll do interleukin 1A because that's actually, or it's
alpha.
Yeah, alpha's fine. So you see, there's all the different ways of writing it, and even that's not exhaustive. If you go to gene cards and then look up interleukin 1 alpha, it'll give you, twice as many other ways of saying it. But anyway, that's just something to think about, to be able to type in and do the query expansion based on gene cards or the mesh term or whatever happens to be handy.
Well, where I can see that being programmable easily is maybe on here. So you could take a term and then hit a button that says expand by blah, and then all the other blahs would end up here.
Yeah.
And then so the other thing too is that you can like send to log. So I could select, oh dear. I don't know what okay, so I can select, okay, I don't know what's going on there. I can, if it works, I don't know why it's not working. I can select a bunch of cells and send it to the log sheet. And the nice thing about the log sheet is if I have a whole bunch of queries here, I can run them all in batch and then they append to each other. So you could have all of your interleukins here, go get all the records, might be too many to run, but and then.
But it can be done in the original PubMed query, right? It's parentheses IL-1A or
True.
interleukin 1A or da da da. So that's, I mean, that's where it'd be nice to be able to do an expansion. But yeah, put it on a tab and then they can copy and paste it over here into your query tab. That'd be handy. But this is really cool.
Okay, do we have time? I have one more thing on my list
Yeah.
to show you.
Sure, we officially could go to 4:30, although, but there's a single speaker. Sometimes people will leave at 4 because, but go ahead, please.
Okay, so the last thing on my list was, I'm just gonna, was, okay, so you have built your pathways and you know what your players are, and then you want to see if there are chemicals that are known to disrupt this biology, and then you can dig in and figure out which, you could actually do a query that just looks at your, your genes, but I'm going to do a query here, blood-brain barrier drug effects. That means that's the way that MESH annotates things that mess with the blood-brain barrier that are chemicals. And then I added toxicity just to bring it down to a manageable level. And I think there are 2,000-ish here. Yeah, oh, just over 1,000, sorry. Okay, so I've got this corpus, and I'm going to do this again with the MESH substances, and I'm going to get major topics only because I have found that that's the best way to find chemicals that are the major topic of the article that might be causing whatever the toxicity is. We used this capability most recently in a project we worked on about the retinoid system, where I took a bunch of different targets in the retinoid pathway and looked to see what chemicals were annotated out there. Now, it won't pick up things that are, if you do in vitro testing of 30 chemicals, it won't pick up every chemical, but it does pick up discussions of 1 or 2 chemicals. So I'm going to go through that process I went through before, get rid of families just because they don't help any. And then I'm going to look at just organics. Now, before, remember, I looked at genes and proteins. Now I'm looking for chemicals. So I'm going to sort on organic. That's the most likely for small molecule organics. And so here are the chemicals that are most annotated in articles about having drug effects on the and toxicity of the blood-brain barrier. And you can see that, they aren't very nice chemicals as a whole. And to see the articles, you can just double click mannitol, about cocaine, and it brings you over here where you can read the article. So, and then you can do the same. Once you've investigated your chemicals, you can pick the ones out that you think are most appropriate for your whatever you're doing and copy them and paste them to, okay, fine. Copy them and paste them to the landscape sheet, where again, you can look at their intersection with genes and with diseases. And let's say you're interested to see, this is blood-brain barrier. So maybe you're looking at autism, and maybe you're looking at Alzheimer's, something like that, with your chemicals. So this would be your adverse outcome. Maybe maybe this is the adverse outcome you're interested in, or you just want to see what the literature says. But so the cycle here is you found chemicals that are known in the literature to have some effect on the blood-brain barrier and have some indication of toxicity. And now you're seeing what the relationship is between those substances and some maybe diseases of interest. And then you can dig into the pathways and so on. Extend your queries any way you want to from here. So that was the 4th thing on my list I was going to show you. So I can check everything off my list, and I've learned a lot today. So thank you for everything. And are there more questions?
Nancy, can you pull the structures in so they can see that?
Okay, that's 7.3. Yeah, so when Tom is talking about a new functionality that has not yet been released to the public, where I could have gone out and gotten things like the DSS Tox ID and the structures and the smile strings, any we're building a bunch of APIs here at the EPA that will let you take a chemical. So we could send the APIs chemical names, and they send back identifiers like our DSS Tox ID. And then once you have the DSS Tox ID, you can send another API call and go get structure, smiles, PubChem identifier, and all sorts of other information. But that's not. I can't even show that outside the environment of the EPA right now because it's not ready yet. But that's coming. So keep checking that download area of the dashboard for the new abstract sifters, because someday it will be available to the public.
Thank you, Nancy. Great presentation. Does this by any chance work on Office 365 online?
Yeah. So I always tell people to download it to whatever and not try to run it in a team or what. I don't even understand this new Office environment because everything seems to be sort of on the cloud. I don't think it works there. I always tell people to download it to their drives, even if drives are kind of virtual nowadays, but and it doesn't work on a Mac either because the Mac doesn't support a lot of this stuff. So
and any plans in the future to make it compatible with the online version? And also, what about like LibreOffice or the open source version of Office? I'm assuming that won't work there.
no, I don't have any plans for that. And quite honestly, we just moved to this 365 thing and I don't quite understand it yet. And so maybe when I understand it better, I'll have a better idea of what can work and what can't work. Yeah. Sorry.
No, thank you. This is great.
I can tell you that this works much better on my home machine than it does on the Office machine that has all this fancy stuff. So, and all the cloud stuff. So
I could ask questions all day. So people can tell me to shut up eventually, but in your PubMed query, can you do authors?
do what?
Author names?
Oh, sure. Yeah. Mhm.
So
yeah.
you add AU after that. So if you had a group of people working together or a giant group of people that were just claimed or just got together and you wanted to figure out if they have any overlap.
Yeah. So people have done that on the landscape sheet too. I had somebody who was like putting authors down, column C and then publication years across the top and looking at author publication year sort of stuff. Yeah.
Yeah, because interesting thing, we've had a couple of, in the last 2 years, situations where you start up a new team that has dozens to hundreds of people. And you'd like to kind of analyze all those people's publications to find synergies and overlaps and things like that. And there really are no good tools around for doing that.
So yeah, I would do like here and then put
The author tag.
I mean, I guess you can use first and last name, and then have.
Well, yeah, again, like you said, sometimes you just have to go back to PubMed and look at their rules for writing queries.
Is it first on whatever it is? Yeah. Yep. That's
Or if you put Glazier up there instead of Knudsen. I don't think Tom and I have any papers.
There you go.
Yeah. But to also be able to cross-reference based on terms, other than, do liver and a group. Oh yeah, that's really cool. That would be really handy. So that also means you could do a query on a mesh term.
Oh yeah, absolutely.
Same thing. You just need to put the mesh tag. Yeah.
When I did the blood-brain barrier, that was a mesh term.
That was a mesh term.
Mm-hmm. Yes.
Okay.
Oh, there's digital twins.
Yep. No abstract.
It
literally says no abstract.
No
well,
abstract.
it's not a scientific paper. There is no abstract. It's an opinion piece, so it doesn't have an abstract.
Any other questions?
Well, I'm still going to have to try to figure out how to enable the macro so I could get it to run.
It's usually asks you a question right up here when you download it, a couple questions, depending upon your whatever.
It said macros are now blocked. And then it sent me on a rabbit chase to try to.
You can do on the file properties. You just check a box there.
Okay, I'll have Joel is the expert, so he's using it. So yes, the developer tab. I turned on the developer tab. Anyway, I'll have Joel teach me how to do it because it looks very, very intriguing, and we definitely are. I mean, I think one of the things we're always interested in is linking functionalities between systems. You have some, as again, the kind of thing I was talking about. You have some experiment that's reporting differential expression in two different pathways, and you'd like to have some understanding of how they're connected. Mechanistic. And so finding that, I think, is really helpful.
Yeah, unfortunately, you still have to read a lot.
Well, I wasn't expecting a magic. I've learned that magic is in short supply. We had somebody from Seattle, from the Systems Biology Institute in Seattle, promised magic that read the literature and reconstructed pathways from the literature without humans doing anything. And I thought that was intriguing, but maybe too good to be really true.
Yeah, the thing that we've found very useful is to go to the mouse genomic site and get the ontologies for different phenotypes and genes, and use that as a basis for bringing it back into the abstract sifter and searching. It's really been a very useful thing, and the more you work with it, the more information you get, and it just seems like it's a never-ending cycle. That's the problem. But it's definitely, as you said, James, it's a way of really picking out information you may not have found by just a targeted approach looking for literature.
Yes, Nancy, this is very powerful. And I understand that you build it on Excel since you're using a lot of tools that are already done, right? Because it would be extremely hard to build everything from the beginning. But there is any plan of like disassociating with Excel in the future?
Well, we do have a very simple version of the abstract sifter on our CompTox chemicals dashboard, but it's very rudimentary. Yeah, that's an interesting question. It's so much easier to develop in Excel than it is to develop in a web stack.
Mm-hmm.
Where you have to have your database person, then your end user person, then your middleware person, then you're this person.
Yeah.
It's much easier.
Yeah.
So
the team of one or two people become hundreds easy.
Exactly. And then you have to have processes to make sure there's quality. And then you have to have, oh my gosh.
Yep.
So yeah, the Excel version is zooming ahead, and the web version is just limping way behind. So
I've got a question.
yeah.
There might be a tool available for this already, but it would be really nice when you're in the box where you enter your query, if you have one of those really, really complex ones with multiple terms and lots of parentheses, if you could enter it in a formatted way with line breaks and indentations so that you can see the logical structure of it, and then have it retain that. So when you click on the query and it pops up, it's still in that format and you can edit it that way. Can you do that?
I don't know. I've never thought about that. You are absolutely right that sometimes queries can get so complex, and there's a lot of legacy queries here at the EPA that people have been using. They think they're perfect, and they pass them on to other people, and they're paid. They're like a couple pages of query. So
and it would be nice to even take one of those and have it do what's called the beautification, where it just relays it out in that
parses it and makes it visually nice. Yeah. So maybe that's an idea that other people at the EPA would cotton on to.
Yeah.
I always say just do a simple query, and then use term mapping or sifting to find what you want. But yeah, people are attached to complex queries.
But
I
to
found.
Sharon's point, would it be possible to say, convert these complex queries into some type of a workflow diagram?
Just
using
to see the
PowerPoint or
structure. Yeah, that's interesting idea too.
It's a common operation. If you Google JSON beautifier or JavaScript beautifier, you can find tools that do that. And I've even found an Excel function beautifier for really complex nested functions.
Oh, I got to find out because there's nothing
Yeah.
beautiful about my code. But
Yeah,
I have
but
seen the
I
JSON ones.
I tried Googling PubMed query beautifier and didn't come up with anything.
Oh, that's fascinating. Yeah, and just one misplaced and or can really mess things up.
Yeah, I mean, the power of it comes across clearly. It's a little intimidating. And
Well,
as somebody who teaches software, I realize that it's easy for you and that it looks, you think, oh, this is simple. And then the audience says, my goodness, that's complicated. On the other hand, Joel started using it and is learning it very quickly. So I suspect that the key thing is to dig in and do it.
Well, in the user guide, it just absolutely, it's like 45 pages. Then it walks you through step by step with lots of screenshots. So if you go through the user guide ones, and there are lots, the README sheet of the Abstract Sifter has links to lots of different videos that you can watch, but definitely just using it and then, yeah, using it is the big thing. Is that what you would say, Tom? Just spend a couple hours on it?
Yeah, I think, you know, you're right, James. I mean, I feel intimidated. I always call her because, you know, I could sit and take 2 hours to do something that she can do in 10 minutes.
Well,
What's the obstacle to having it work on Mac since Excel works on Mac?
Excel does, but a lot of these the code behind the scenes is using libraries that have not been written for the Mac. So the Mac just doesn't support. So my method of writing programs is if somebody else has done something and it's out on the interweb and they give you the code, I will take that code and modify it. So, and a lot of the times that code uses libraries that are just not supported by the Mac. So that's all I can tell you.
So I take it you're coding in what is it, Visual Basic?
Yes.
But maybe you're importing things that are written in something else.
Well, so there's some libraries, and I assume they're all binary. There's a lot of, I don't know how to talk about it. There's libraries where functions that are in libraries, you just call them, like the API
They're just
stuff.
compiled.
Yeah,
Windows.
and they show tell you how to use them, and you just use them. And so some of those things are very specific to Windows. The APIs. Now I'm working on this section, this code that will, in the notes section where you can get your PDF and extract tables from your PDF. So, and the way that works is that Excel opens up your PDF in Microsoft Word and uses built-in Word things to extract the tables from it. So it's very much a Microsoft environment. And sometimes there are just some things that don't work in a Mac. So,
but all this Microsoft stuff works on the Mac.
well,
I'm just
and also
complaining now.
I am not a Mac person, and we don't have Macs at the EPA. So, you know, if somebody wants to rewrite it for a Mac, they can do that. Sharon.
I'll keep it in mind.
Absolutely.
Okay, I think we've had a great discussion. Any other comments or questions for our speaker? Thank you thank you again for the demo, and appreciate your patience in trying out the terms that I gave you.
Oh no, that makes it fun for me.
See how that works, and I'm sure we'll be using it, and at least the people in my group will be using it and then teaching me how to use it.
Thank you all very much. I really enjoyed it.
Okay, thank you, everybody. We won't have a meeting next week. We'll see you in 2 weeks. And
I have a short question if there is time.
oh, Idemar, go ahead.
And I have to admit that I didn't listen to everything because there are several other things happening. But my question is, because I didn't hear anything related to my question, can you use NLP and augment it with some kind of tricky algorithm about association? Namely, if I'm looking for something that James wrote, I may associate some people who know James, and this way I can get a subgroup of concentrated type of abstracts.
Okay, so we did go, did you see us go over the author stuff, the co-occurrence of authors? Were you watching when we did that?
No, I'll have to go through the recording.
Yeah, go through the recording. There's also one other feature that I did not show, which is, I believe, written about in the user guide, that when you're on the abstract sheet, you know, where you're just looking at one abstract, there's a button to the top that says like this, comma, or question mark. And that lets you, so PubMed has this capability to find articles that are like the one you're looking at. And it uses some text mining algorithm and distance metric, et cetera. And it goes out and finds the 200 articles that are most like the article you're looking at. It can also go find all the articles that have been cited. If the article in the abstracts is in PubMed Central, you can get all the other cited articles, and then you can get articles that cite this one in PubMed Central. So that's one way to do sort of citation chaining, which kind of sounds like maybe you're getting to. So those capabilities are there too, but definitely if you're looking for co-occurrence of authors, something like the landscape sheet would be a good starting point.
Okay, great. I may come back with other questions, but you see the lawyers are using NLP extensively or intensively, whatever, and they may collect similar precedent cases via NLP. And that saves them a lot of reading and flipping pages. So I'm thinking about something similar in your abstract sifting, but I may be wrong, of course.
Well, if you could write down that question with regards to a biological question, I could think through it and help you out with it.
Okay, cool. Thank you.
Thank you.
Okay, well, thank you again for the extra question and appreciate you coming. We've used up our hour and a half and we learned a lot. So we look forward to maybe having you back and giving us a tutorial again in the future. Especially if some of the people start using it, I'm sure that there'll be lots of questions. So appreciate it. So again, no meeting next week. We'll see everyone in 2 weeks. And thank you so much for being willing to speak to us.